Hi, everybody. I see you all. We're waiting to allow a few other people to join. We'll give it a few other more minutes, and then we'll start.
Jonathan.
Call my mom. Okay, I think we're gonna start. Hi, everybody. For those who don't know me, I'm Wendy Aviv, Five Senses Tour, and I'm thrilled to be able to have this little event on our casual Sunday afternoon. Just a little blurb about the five senses. We take tourists to Israel. We are not taking tourists to Israel because nobody's taking tours anywhere right now. But we do hope for that to happen in the future, to go back to some type of normal life. And we're actually going to be adding Dubai when we can fly. So that will be an exciting addition. Absolutely thrilled to have Professor Gershoni. Before we get to that, though, there are just a few housekeeping. I have been promoted today to Chief Technology Officer of the Zoom, not to one-up my husband, but so if anything goes wrong, just bear with me and I'll make it go right again. So just be patient. As you can all see, this is a Zoom meeting and everybody has the option to speak, but in order to make it so we're not talking over each other, I have a bunch of questions. Please make sure you're all on mute. And if you want to ask something, you can put it in the chat and I'll see if we have time to get to it. We've sort of, the questions that I've gotten have sort of been worked into like 4 categories, more or less, which are obviously the vaccine, treatments, questions about testing, the validity and all those kind of things, and the actual illness. So without further ado, let me introduce Professor Jonathan Gershoni, born in Israel, received his PhD in biochemistry from the Hebrew University of Jerusalem, postdoc training at Yale School of Medicine, and for 30 years he's been studying the immune response towards viruses such as HIV, HCV, SARS, COVID. So plenty of experience there before we ever knew what COVID was. Worked as a visiting scientist at the National Institute of Health in Maryland and at Boston University Department of Physics. As the researchers around the world are racing to get a vaccine and drugs to help the virus, Professor Gershoni was awarded the US patent for the virus vaccine design. On a personal note, I'm thrilled to be able to meet him. Professor Gershoni's parents and my parents have been dear, dear friends, and this is the first time I've been able to meet him. So your dad and my dad are looking, saying, what the hell has happened to this world? And they're laughing and saying, at least something good came out of 2020. So without further ado, I look forward to meeting you in person in December when we head to Israel. Without further ado, please start us off, and then I'm going to ask questions.
Okay, so first of all, this is an amazing opportunity. I am delighted to meet with you, Wendy, and Kathy and the family. This is a reunion in cyberspace, so it's a bit bizarre, but we will make up for it, and we'll see each other certainly in the not too distant future if we look at how things are developing. A few words about my research. I've been looking at the interaction between viruses and the immune system now, as you said, for many, many years. And I was giving a lecture in Boston describing research that we were doing on HIV/AIDS, which is something we'd been doing for many years. And one of the participants in the lecture asked me after the lecture to join him in his office because he wanted to describe a virus that he's currently working on and felt that this might be an opportunity to collaborate and apply some of the technologies that we were developing in studying the virus that he's interested in. And this was in 2004. And this was SARS coronavirus 1. And that's how I was introduced to SARS. So I've been studying coronaviruses, which is a family of viruses, now for 15 years. And it's quite rare and unique because I have the sense that I've been working in this field, trying to figure out ways to develop vaccines and so on for 30, 40 years. And now all of a sudden, I've been given a stage and I have the opportunity to try to apply some of the things that we've been doing and hopefully contribute in a modest way to developing vaccines and certainly sharing what experience I have with others. I think that there are a number of points that would be worth clarifying. And again, I don't know the audience and how much people really know. And one thing that makes it important to realize is what is exactly a virus. So I'll take one step back. And you have to think of a virus, for those who are not biologists, as a disk on key, a flash key. We realize that in the disk on key, there's memory inside, and it's within a little plastic box or coating, and it's got a USB plug, and that's it. It's not a computer, but it does contain information. And viruses are exactly the same. They have information stored in a protein little box, capsid, and on the surface of the virus, they have their USB plugs, which are the spike proteins. And when people talk in terms of coronaviruses and the spike, this is very much simply a means for a bit of genetic information to be transmitted into cells via the interaction of the spike, the virus's, if you will, USB plug, inserted into the socket of the computer. And in the same sense that a disk on key, a flash key, is not a computer, viruses are not cells. Viruses are not live. They are particles. And the world of viruses can be divided into two groups. Those whose genetic information our DNA, like we have. It's become a popular term. We all say, well, it's in my DNA, or it isn't in my DNA. So DNA has become a term that people sort of regard as our heritage, our genetic background, and basically who we are. So some viruses have DNA, but others have a different molecule for their memory, and that's RNA. And the flow of information in our cells, in living beings, is that the information is stored in DNA, but a copy of our genes is made in a very labile and just like a Xerox copy of the genes, and that's our RNA. The RNA leaves the nucleus of our cells and is translated into protein. And protein is what actually does everything in our body. So we have to have that threesome. We have to have the original information, DNA. It's made into a disposable copy, RNA, and then that's translated into protein. And as I said previously, viruses have either DNA or RNA, but never both. And so under the circumstances, viruses are simply not alive. And so they cannot replicate. They can't cause us any harm so long as they don't enter our cells. And if, on the other hand, the viral USB plug, that spike, binds to the receptor, the socket, if you will, on those cells that the virus is going to infect, then the genetic information in the virus can enter the cell which it infects and then make proteins and whatever in new viruses. So this is, I know it's not anything specific for COVID-19 or for coronaviruses. What I've said is true for all viruses. But before we start talking in details in anything to do specifically with COVID-19, which is the disease, and SARS coronavirus 2, which is the virus which causes that disease, in the same sense that HIV is the virus which causes the disease AIDS. So these are two different things. It's a matter of semantics. Then we have to realize, first of all, if I summarize, viruses are not alive. They contain either RNA or DNA. They infect cells through the interaction of their spike on the opposing cell, which is alive, and it's got the socket, so to say, the receptor. And it's only through this specific interaction can a virus infect a cell and then replicate within the cell. And so those are very fundamental concepts that we need if we want to talk about anything to do with SARS coronavirus 2 and COVID-19, the disease and vaccines, if you will, diagnostics, and so on. So I think with that background, that gives you the option then to ask a question.
So, okay, so that's going to move me to a different question as my first question. And it's, I guess we all wonder when you talk about it in that format. So not everybody is equally susceptible to this virus, because I'll give you an example. My son was with a friend of his sitting at a restaurant, windows were open, 3 hours. He was the next day figured out he had COVID or tested positive. Yet my son's been exposed multiple times, has tested multiple times, and has never gotten it. So I guess our DNA determines how susceptible we are to that virus. Is that?
Well, first of all, your son's lucky.
Okay, good.
That's cool. And we have to bank on luck. Okay,
Yes.
we don't take it for granted. In reality, I'm not sure there's any real substance to the suspicion that your son, for whatever reason, seems to be less prone to getting infected. Meaning, does he have something which gives him the sense or the ability not to get readily infected, whereas other people do. There could be, I don't know. However, you're absolutely right in saying that for various viruses, some people are more or less susceptible. And in the case of AIDS, I don't know if you recall, but it was discovered that there are some people who have a mutation, and this mutation of 32 base pairs and so on, you don't care about the details, but made them, for the most part, resistant, and they don't get infected with AIDS. And that's pretty cool. But that is based on the fact that in order for the AIDS virus to infect its white blood cells, there has to be compatibility with the spike on the one hand and the receptor on the other. And if for whatever reason, some people have a different composition in receptor that makes them less prone to interact with the virus, that could be one reason. There could be other reasons that for whatever reason, and I have no idea, maybe the virus is less efficient in infecting if, let's say, the saliva or the secretions in a given person's body might be a little more acidic, maybe a little more basic, maybe less viscous, more viscous. These could be things which would be secondary and may turn out to provide an advantage to some people. We don't know enough about that. So whatever the situation is, obviously your son's lucky. He should not chance and be flippant about the fact of not taking care because he's resistant. We've seen this happen in many, many diseases and situations. Some people, discordant couples, where one partner is HIV infected. They've had obviously unprotected sex if they are a couple for years. And for one reason or another, this particular other partner simply does not contract the disease. Okay. Why? We don't know.
Okay. Okay. I'll tell him to go play the lottery since he won, and
yep.
to stay put. Okay. So vaccine, that's what everybody wants to know. If you had to make an educated guess based on all the information as we know today, taking into consideration treatment, vaccine, number of people willing to take the vaccine, how long do you anticipate how long do you anticipate that it will take life to get back to pre-COVID normal of travel, companies calling their employees back, really full on?
Well, I think there's going to be a post-corona normalcy. So I don't think that we need to think in terms of at one given point, we're going to be back where we started in November of 2019. That's never going to happen because the world has changed. And so here we find ourselves zooming and adopting various behaviors and conceptions that are going to change. I've visited the Far East numerous times, and I've found them wearing surgical masks as being rather peculiar, hypochondriacs, and not fully understanding how they continue life with such this, I mean, coming from Israel or and seeing groups of Japanese tourists or Koreans and so on, everybody wearing a mask, we regard this with a level of arrogance and disbelief. And I think that now post-corona, we will look at it differently. I don't know. It might be the case. I'm asthmatic, and I have not had asthma since corona because we're all wearing masks. And so I'm probably not exposed to as much pollen and pollution. And so, who knows? There may be good things that have changed our way of life. However, to your question, when are we going to get back to some level of normalcy, the ability to travel, to work, to go to school, to hug and kiss our loved ones? And obviously, this is still going to take some time. It's going to take time because people are going to have to begin to seriously understand that we have to vaccinate. I think there's a basic misunderstanding about what constitutes herd immunity. The concept is rather simple. If there are enough people out there who are immune to the virus, then a virus which emerges from an infected individual can't find fertile ground to infect others because everybody around him is protected. And that's the concept of herd immunity. Now, in order for herd immunity to be effective, we need sufficient coverage of about, I really don't know where all these theoretical biologists come up with easy numbers. We don't need much more than 50%. I do not understand how you get to herd immunity with much less than 80 or 90 and preferably 95% coverage. That is what we see for measles. That's what we see for most infectious diseases. We need to have at least 80% of the population immune to the virus. Now, another misconception about herd immunity is when you hear people who argue that the way to handle with the epidemic is to allow us to achieve herd immunity through natural disease. And that's basically very dangerous because it is true that if somebody is infected in a natural situation, he is immune. And if everybody's infected, then the virus is confronted with the possibility that he can't infect. But in the wake of everybody getting infected, you're going to have 1%, maybe 2% of the population die. And so you just have to think about it. If in Israel we have 300,000 people infected, let's assume that number is an underestimate. Let's go crazy and say that in reality we have 1 million people who've been infected. We just don't know it. But 1 million people is only 10% of the population. We're a far cry from 80%. And if we believe the numbers of how many have already died, well, we're close to 3,000 people who've died. And I don't want to get to natural herd immunity witnessing 30 to 50,000 people dying. And so, whereas in theory, herd immunity can be achieved through natural infection, that's a pretty pitiful way of going. Whereas in contrast, if we implement vaccination and vaccination campaigns, then we can see that we'll be able to achieve a high level of protection of the population and society. And we would like to try to reach around 80% and more. And then we would have effective herd immunity and we'd be able to get out of it. And so this is what we're heading for.
So the $10 million question then is,
Yeah.
While the vaccine, obviously everybody's in a race to get a vaccine and they're fast tracking, and I assume the fast tracking is cutting through red tape, that they're not on the bottom of the pile, they're on the top of the pile through all that stuff. But is there a medical technology that can fast track potential side effects 10 years down the line? How do we, I think of before my time, but I think of the thalidomide babies and what happened with that vaccine. So what is the logical rollout to ensure safety with with all the components that everybody has to think about? How do you fast track knowing what's going to happen down the line?
Okay, so first of all, I understand, and I think there's a bit of misconception as to what's fast track. If we go back to the 1940s, people were confronted with a terrible paralytic disease called polio. Now, it wasn't quite clear what the causative agent of polio was. You have to realize that we're confronted with a disease and we know it's contagious, but we don't, until we physically isolate the etiologic agent, the cause of the disease, we don't know. And then once we've been able to isolate it, then we have to characterize it. And this takes time. And we have to figure out, maybe we can't even grow it up in the laboratory. And so you have to figure out how to grow the virus and how to characterize it and how to characterize the disease. So if you think about that, in polio in the 40s, it took John Enders, who got the Nobel Prize for this, how to define the virus and learn how to grow it up and characterize the polio virus. It then took Jonas Salk time to figure out how to grow up enough of the virus and inactivate it chemically with formaldehyde, and then figure out that if he injects animals with this inactivated virus, it'll be effective in raising an immune response and protection. So from the early 40s and up to April of 1954, this took a long time to understand polio. But it was in April 1954 that they started the phase 3 clinical trial. So everything preceding that took a long time. But the phase 3 clinical trial started in April 1954 and it ended in April 1955, 1 year. And so when we're talking about, we're warp speed and so on, basically, and I'll relate to that in a second, once you get to the point where you know what is the virus and how to deal with it and how to generate your vaccine, then the clinical trial takes time, but it doesn't take an enormous amount of time, especially if people are getting infected. And so that allows us ethically to run a phase 3 clinical trial with placebo and nonetheless get enough people infected so we can see the effect of the vaccine. And so that's cool. The day in April, I think April 26 or whatever it was in 1955, that the results of the polio vaccine, Salk's polio vaccine, was revealed and declared a huge success, the following day, FDA had licensed the vaccine for mass production to 5 independent industries. So, we get all hung up now about this fact that they're fast tracking. Is that dangerous? I think a lot of that is anxiety, which is fair enough. People are totally stressed out and for the most part tired and wary about what's been going on. And they assume that we have to follow the same timeline that was unfortunately the reality 50 years ago. But we're not there anymore, in the same sense that we're not surprised you can fly to Tel Aviv and Dubai much quicker than the Wright brothers had thought about in Kitty Hawk in 1903. Technologies have changed. And so if we now look at what has been the course of COVID-19, SARS coronavirus 2, the Far East had been acquainted with coronaviruses as of 2002, because that's where SARS coronavirus in Guangdong had emerged for the first time. There was an epidemic. People realized, okay, this is how you deal with epidemic. You wear masks, you social distance, and they learned about coronaviruses coming from bats and the impact in wet markets and so on and so forth. They culled tens of thousands of civet cats, which was the intermediate vector. And so the people gained experience. And then in 2012, yet another coronavirus emerged, MERS. And again, people realized, okay, we understand that. We know the molecular biology. We've analyzed in molecular structure the genetic composition. We know where the spike protein gene is, and we know the biology of coronaviruses. And so for 20 years, people were well prepared to isolate and analyze coronaviruses. This is not new.
Mhm.
And so if in 2019, a coronavirus appears in Wuhan, then we have all the technology. I mean, we've isolated HIV and HCV, hepatitis C. We know how to deal over the past years and years and years, 30, 40 years of experience in how to culture and analyze and characterize at the molecular level viruses. And so is it a remarkable achievement that if in 12, I think it was 12 of December 2019, that there were the first indications of an unknown disease. And by January 9, the Chinese published the complete genetic composition of what is today SARS coronavirus 2. Now, people who talk about conspiracies and whatnot, I cannot really understand why people are arguing that the Chinese withheld information. We're talking about the first indication of an epidemic was in the middle of December, and January 9, I personally had the complete genetic composition of this suspect virus. Now, anybody, any of my students could look at the genetic composition and say this is a coronavirus. And not only that, they can easily pinpoint and say, from here to here, this is the gene that corresponds to the spike. And so this is just because we've had years and years and years of experience. So now the question is we know what the target is. We know it's a coronavirus. It didn't take us years to discover that. We know it because this is just plugging it right in. And then we know exactly the composition of the spike protein. And everybody and his uncle immediately started to develop vaccines because we know the paradigm for a vaccine is use the spike protein. That's why I said, let's invest a few minutes to understand that the Achilles heel, the soft spot of the virus, if we want to knock it out, is we have to raise an immune response to the spike. And this everybody knew. So now all of a sudden you have the whole world trying to develop various technologies on how to exploit that knowledge and the understanding that the ultimate target is the spike. And how do we translate that into a vaccine? Now, there are various ways. If we take all of the vaccine modalities and decide to divide them into two types of vaccines. So this is the way I think about it. It's not textbook, but conceptually, I think it's useful for the general public to think about vaccines. Vaccines can be of two general types. What I refer to as hardware vaccines. And a hardware vaccine is any vaccine that has the physical presence of the virus material. So it could be taking an intact virus and chemically modifying it so it becomes a killed non-infectious virus, as is the case for polio, inactivated polio vaccine of Jonas Salk. So we've done this. This was 1955. So we know how to do that. So we can cultivate the virus and inactivate it chemically. That would be one approach. And that would be hardware because it's physical presence of the actual viral proteins and material. The second would be let's just synthesize in the laboratory just the viral spike, what we refer to as a subunit vaccine, but it is the protein of the virus. And we do this all the time. Every baby in Israel who's born, before the baby leaves the hospital, it gets a shot of the surface antigen, the coat protein of hepatitis B. We don't inject the entire virus. We don't have to inactivate it because the protein is not dangerous. It's just pure protein of the spike protein of hepatitis B. The surface antigen, that's the subunit vaccine. That is a hardware vaccine. It is the physical protein of the virus. So everything we've learned to love about vaccines are hardware vaccines. That's polio, or that could be Gardasil for cervical cancer, and it could be hepatitis B. And then there's the live attenuated viruses, but they're also hardware vaccines. Those are the viruses that have been modified not to cause disease, such as Sabin's polio vaccine and such as measles. Influenza is killed. So we have these are all the vaccines we know. And then the other class of vaccines is what I call software vaccines. Software vaccines are vaccines that don't actually deliver the physical composition and antigens, the proteins of the virus at all. But that's why when we started this conversation, I said, I want you to understand viruses contain the protein, but viruses contain the genetic information inside, RNA or DNA. And so a software vaccine has taken a brand new approach. It's new technology. The software vaccines say, I'm not going to inject anything to do with the proteins of the virus. I'm just going to inject the genetic information, the software. The instructions is how to make the protein. And that software is I'm going to take RNA of the virus. Coronavirus is an RNA virus. I don't need all of its RNA. It's very, very large. I need a snippet, 12% of the virus. That's all I need in order to code for the protein of the virus in the body of the person who receives the vaccine. So we inject not the protein, but rather the gene that is responsible for producing the coat protein. So this is a new approach. This has never been done before. This is an RNA virus, a vaccine, and this is what Moderna is doing. This is what Pfizer is doing. This is what Arcturus is doing. There are many companies that now have said, we're going to use a software vaccine. But there has not been one software vaccine thus far which has been developed, phase 1, phase 2, phase 3, approved, licensed, and used in the public. Zero. So this is a new generation in developing vaccines. It's much easier. It did not require how to figure out how am I going to grow the virus? How am I going to upscale into billions of doses? I'm just going to inject the software. Now, the same is true, it's a slightly different approach for Johnson & Johnson, AstraZeneca, and Gamaleya, the Russian vaccine, for example. There they've said exactly the same. We're going to inject software. We're not going to worry about trying to produce in mass the virus. What we're going to do is we're going to take the viral gene for the spike exclusively and only, and we're going to package it in a other virus which is harmless to people, and it's going to deliver the viral genetic information into the arm of the vaccinee. And that other virus, which is not harmful, does not cause disease, will enter our cells, but it won't replicate. It will be a non-replicating virus, and it will deliver the genetic information, and we'll get the same effect. We'll get production of the spike protein in our tissue, and that will elicit the immune response. But this is, again, a software vaccine. And there are no approved software vaccines as yet. So we're on the verge of a new generation of vaccines. And we have to be brave about that in the sense of critically understanding what these vaccines do. But because they're new, does not necessarily mean that they are super dangerous or we've cut corners. It's a new approach. In parallel, there are a number of companies, Novavax and Sanofi, which are developing the subunit vaccine, just the protein, the spike. And there are at least Sinopharm and Sinovax and a bunch of others that are doing inactivated virus in China and in India. And this is going a bit slower, but this is the natural course, using the traditional vaccines on the one hand, and they'll come through, and this new generation of vaccines, which is much easierly scaled up and much faster and safer to produce.
Okay. So there are a few other questions, and I know our time is winding down a little bit. So a yes or no? So you would have no concern taking one of these new types of software vaccines? You feel they are safe? Yes? No? Yes?
Yes.
Yes. Okay.
I don't think that RNA is dangerous.
Okay.
It doesn't replicate in our body. It's localized to the area of the injection. And it does not modify genetically the cells that receive the RNA.
Okay.
And if you think about it, I've said that the coronavirus is an RNA virus. A person who is infected by the virus contains not 12% of its genes, but all of the genes. And the virus replicates frantically in mass. So the total dose of vaccine that you get from coronavirus, from the infection, is orders of magnitude more. And the encouraging aspect is the vast majority of people who are infected recover.
Okay, that's a whole other conversation. So, switching gears to testing. What is the proper incubation period before you get testing? Why are there so many false negatives, false positives? Rapid test versus the 3 to 5 day test. What would be your recommendation as far as really a protocol to follow?
Okay, so now we're getting into a field where I have some experience, but I really haven't been looking at COVID-19 per se. We know that from the moment a person is infected, it takes time before that person starts to accumulate sufficient quantity, physical quantity of virus, which falls within the range of sensitivity of the test we want to use. Now, in the world of HIV testing, we normally look for antibodies against the virus. This is the blood test. And it takes a few weeks before we start to begin to accumulate a signal, and as much as a few months. So as you indicated, there is a window of uncertainty. I was infected in day 1. And if I'm going to do the serological test, that is to test the presence of antibodies, I need to be able to raise an immune response and accumulate enough antibody in my blood so as to be detected by the blood test, which has to have a certain level of sensitivity. So
But
that could take weeks.
That's after you've already been sick and you've gone through the...
In the case of COVID-19, you would probably recover, okay? So this is the blood test, the serology.
Right.
And the serology goes to say anybody who is positive in the blood test is a good indication that he had been infected. And if he's feeling well, he's recovered. It gives us an idea about the numbers of how many people have actually been infected, but it doesn't do you much good in controlling the pandemic. Okay, so what you would like is you would like to have a quick test which says, I was infected today. I would like to know that tomorrow. I don't want to have to wait a week or so. And the question is how much time does it take for an infected individual to start to accumulate enough viral particles, the actual virus, and we don't see the virus in our blood. The virus is actually accumulating in the mucoid secretions of our nose and oral cavity. And so this can take I would expect a few days. I don't know the numbers, but I'm saying this will take a few days because what we're to do is actually measure the physical presence of enough viruses to give us a strong signal. Now, the methodology is quite brilliant, and the person who developed that years ago got the Nobel Prize for polymerase chain reaction, for PCR. And this technology has a unique characteristic in taking even minuscule amounts of the viral RNA as a template and then amplifying the signal very, very, very much, orders of magnitude, to bring it within the scope of detectability of our machine. And so that's the fundamental test. We're testing the physical presence of RNA in our secretions. When you get a good, strong profit, go ahead.
I guess the question is, it's not necessarily I understand there are cases of false positives and false negatives, so it must be related to either the person who's administering the test, how they do it, or
No,
the timing of when the test was done compared to when the person was actually exposed to it.
I think there are a number of factors here. One factor is human error. Let's put that aside. Let's just, for the sake of grins, decide whoever's doing the test, the machines and the technicians are all great. Because we can always argue that there could be some level of incompetence and inadequacies. But assuming everything is great, then we want to look objectively. So as I was saying, if you get a strong PCR positive result, that's positive. And I would not doubt that in a moment. But that takes a few days, and that requires PCR. And what people are trying to do is to develop quicker and earlier tests. So when you get into quicker and earlier tests, you're confronted with the need to test suboptimal amounts of virus. And this could be, as we've said, it could be that insufficient time has been given for the virus to accumulate. There could be errors in where to take the swab. So if it wasn't taken efficiently well, and there was not enough mucoid secretion that contained the virus, that could lead to a false negative. The problem is that now we're trying to speed up and mass produce, and so you have new tests which are based on fluorescence, Sophia and others. And these tests, typically, they have less efficiency, fidelity, and robustness, because we're trying to get an idea, am I within the range? You know, even for HIV, the initial test gives a signal, this is probably positive. But we don't go and call up the person and say, it looks like you're probably HIV infected. What they then do is a second test, and that's Western blot. And Western blot is confirmatory. And so everybody currently is in a frenzy. As I said, we're all psychologically damaged at this point. and we're all trying to cope. And so we're all looking for a quick fix. And it's difficult because we want to know, and we want to know yesterday. this is a problem. Now, part of it is compliance. If I am suspected of being positive, then I should be in isolation. And I don't want to be in isolation. So I'm pushing the system and saying, give me an answer, a credible answer earlier, so I don't have to, we've all experienced our working with computers. So when I started to work with a computer, the download time was, I had to download a megabyte or something, and that could take a few minutes. And now, if we were confronted with downloading a document which is, hundreds of megabytes and it takes more than seconds, we're pissed and we argue, God, this is so slow. And so we've changed the pace of things, and there are no quick fixes. There is not going to be a unique test that is able to give you a credible positive response before the virus replicates to a sufficient level. Now, it's interesting. There could be new parameters, measures of infection, secondary surrogates. So it could be, I've heard some people doing research on the effect of coronavirus on our bacterial microbiome. And if the effect of the virus impacts the microbiome, and if there are bacteria that have different smells, different odors. So people are saying you might be able to detect the presence and changes which correlate and so on. I don't know anything about these things, but the fundamental PCR is a very useful measure. It takes a few days until we get an established signal. And serology gives us a good tool for epidemiology. And we have to realize that there's no miracle out there.
No miracle, quick fix, that's for sure, considering we're going on a year of this. So the last question I have before everybody moves on, when do you think we are going to be able to, the general public, be able to be vaccinated and get back to some sense of normalcy in our life?
Okay, normalcy in our life is split in the sense that once you're vaccinated, you can immediately, within a few weeks from vaccination, maybe we know, for example, some of the vaccines require an initial vaccine and then a boost 3 weeks later. So let's say you complete the vaccination regimen. That should give you a very high level of normalcy and sanity, in the sense that you know that you're, for the most part, protected. And all the anxiety of, am I going to contract the disease, is going to be removed. Am I going to infect others, is going to be markedly reduced and removed. We don't know that for 100% definite confidence, but we're assuming that vaccinated people are not going to spread the disease. And that should be a pretty good dose of normalcy for the average person. The question is when can the country get back to normal? And that is when we gain, I would say, sufficient herd immunity or vaccination depth. And I would like to hope greater than 50-60%. Now, if we look at the projections, Pfizer and Moderna talk about the fact that they should have well over 50 million doses by January 1, which means that 25 million Americans will be vaccinated shortly thereafter. Of course, there's the cold chain logistics and so on and so forth. My real concern is, how do I convince the public to want to get vaccinated? There is so much nonsense in anti-vax and lobbies and people who are reluctant to get vaccinated for no reason, that I find this dangerous and unfortunate. And so the question is what do we have to do in order to encourage people to get vaccinated, embrace vaccines, and implement them. And for this, I think that we're losing time. I think that educational campaigns in explaining the value of vaccination, the presumed safety, there is no way that we can talk about what are going to be the long-term side effects. I can't guarantee that because we don't know. But one can say with confidence that I don't foresee there should be any reason to expect terrible side effects. It doesn't make sense. And so I'm saying that we have to try to inspire confidence in the vaccines and in using them. I think that good examples of politicians and leadership and celebrities and so on should demonstrate a willingness to be publicly vaccinated and illustrate, here, I believe in the system. So I would hope that we're going to see various celebrities or people who give confidence to the general public to say, yes, I've been vaccinated and so on. And then hope that there will be enough vaccines to go out there. My sense is we're going to have a greater problem in convincing the public to use the vaccines as opposed to manufacturing and distributing them.
Interesting.
That's my main concern. I'm trying to do something here in Israel. I've failed gloriously. We're not talking politics, but the politicians have their own agenda, and I'm very, very concerned on the one hand, and equally frustrated in the sense that I don't think enough is being done as far as educating the public to understand that purchasing vaccines and not using them is worse than buying a book and not reading it. So we really have to work on the education and preparation of the public to embrace the vaccines. And that's really important.
Okay. Okay, so I'm based on that, I'm shooting for third quarter of 2021 that we're gonna start.
I would expect so.
Okay, done.
Okay.
Done.
Well, we've agreed.
We
Now,
can-
if people are interested, and I don't know, it's not my position to give a promo or a plug for my course, but I have an online course in edX, which is called Viruses and How to Beat Them. And if people are interested in a little more background on cell biology, viruses, immunology, and vaccines, I put together a course for the general public. It's not for hardcore molecular biologists. And it's free audit. All you have to do is look to edX, EDX, and my name, and it'll pop up, viruses and how to beat them. And so it's a systematic course for the general public, because I sincerely believe that a major obstacle is going to educate the public and inspire confidence that people will want to get vaccinated. And that's important.
Okay. Okay. Fabulous. I'm 2 minutes over. I want to stay true to my timing. I cannot thank you enough for taking your time, your Sunday evening, and sharing. And I recorded this, so I will send it out in case people weren't here for the whole thing. They want to share with other people. And you're somewhat convincing me to open my eyes to getting a vaccine. So you've moved the needle a little bit.
That's good.
You've
I
moved
think
the
people
needle.
should simply get vaccinated.
I think I just want to travel so badly that I'm ready to just
I
go
honestly
for it.
believe I'm totally against compulsory vaccination. And I do believe that one of the ways of increasing vaccine coverage is to link vaccine with travel. And in the same sense, when I fly to Kenya or Uganda in Africa,
Right.
I'm required to take a yellow fever vaccine and Japanese encephalitis and other vaccines when I travel to the Far East. And we don't think twice about it. We basically accept the fact that if I want to travel, this is something I have to do. And so for Israel, I think that what we basically need to do is say, you don't have to have a vaccine. You can stay in Israel. But if you want to travel, you cannot leave the country without a stamp that you've been vaccinated. And you can't re enter the country without being vaccinated. And so this is up to you. And I think that that's a pretty good incentive to enhance the coverage of vaccination in a given country. And I think that's a valid point because it is up to our border control to protect the country,
Right.
and that's valid.
Right. Right.
So that's more or less what I believe.
Okay, well, again, thank you, thank you, thank you for everybody that joined, and I will send out a recording, and I appreciate it. Stay healthy, stay well, and let's all hope for travel into 2021.
I'm all for it.
Thank you so
Thank you.
much.
Bye.
Bye.
Love you, Kathy.